as Dr. McEwen mentioned, my background is in surgery, and my research interests are in clinical epidemiology and metabolomics. Metabolomic Technology Incorporated, or MTI, is a new Edmonton-based company that Dr. Richard Fedorka and I started, and it's in a joint venture with Tech Edmonton. And we're looking at developing and commercializing diagnostic tests for gastrointestinal diseases. The science behind it is based on research that I've done for my thesis and my master's. So today I'd like to talk to you about the first product that's currently in development, and we call it ColoDx. It's a spot urine diagnostic test with high sensitivity to detect colonic adenomatous polyps. And these polyps are precursors to colorectal cancer. So colorectal cancer is a major public health concern. It's a major cause of morbidity and mortality. In the US alone, 220 new cases of colorectal cancers are diagnosed a year. The National Cancer Institute predicts in 2010 there will be about 51,000 deaths from colon and rectal cancers combined. So it's a big problem. However, if diagnosed at the polyp stage, or the adenomatous polyp stage, colorectal cancer is completely preventable. The pathway that normal colonic epithelia becomes malignant is long, and it undergoes 2 genetic mutations. So there is time for detecting the polyp at the precancerous stage, and this makes a population-based screening program possible. So, as many of you know, our current screening methods include fecal occult blood testing, flexible sigmoidoscopy, air contrast band minima, or the gold standard colonoscopy. Now, the invasive tests are associated with risk and potential complications, and the non-invasive tests are yucky to do because it involves a stool test, and people are often non-compliant. Although we do have population-based data from the Nottingham trial showing a 15% reduction rate using the fecal blood test alone, even only with 40% uptake. However, the fecal occult blood test for the guaiac-based testings only detects polyps with a sensitivity of 5%, and the newer immune testing types of fecal testing detects polyps with a sensitivity of 30%. So with the low uptake and the low sensitivity in these non-invasive tests, cancer reduction with current screening technology can be challenging. Some of the newer tests are listed here, and some only detect colon cancer and not polyps, and the cost can be quite high. Now, metabolomics is a new field of science that's downstream from genomics and proteomics. So metabolites are to metabolomics as genes are to genomics. So it's downstream, and it's influenced by diet, by disease states, and by genetics. Small changes in genetics can result in large changes in metabolites, and it's used, this is a field of science that's being currently used to study various clinical states like schizophrenia, preeclampsia, and oncology. Now, Alberta is one of the leading, it's Canada's and perhaps even North America's leading center in metabolomic research. Some of the scientists in Alberta have been involved in some of the initial milestones to move this science forward. So like I said before, metabolomics is, or metabolism is affected by genetics, by diet, by disease states. The process of metabolomics involves collecting biofluids, including blood, urine, serum, tissue, and subjecting it to NMR or mass spec analysis where metabolite concentrations are generated. Because there are so many metabolite concentrations, advanced statistical software is required to do the data analysis. Our group did a systematic review recently, and there are only 8 papers in the literature on this topic of colorectal cancer and metabolomics, and only 2 of those studies used urine. Although the worldwide interest in finding a biomarker for colorectal cancer is huge, and some of the recent GI meeting in Europe, there are 2 abstracts addressing this topic, and it's based on some newer technologies like stool DNA and nucleic acid analysis. So our clinical study involved recruiting 1,400 patients, 200 of whom had colon or rectal cancer of various stages. And then through a colorectal cancer screening program, we recruited the other 1,200 patients or subjects. From these patients, they're otherwise healthy, over 50 years old, going for screening. This patient population provided us with our normals controls, who are colonoscopy negative normals, and our adenomatous polyp population. So urines collected prior to the scope and prior to biopsy and prior to removing the polyp. So when the urines are collected, the polyps are still in situ. This provided us with our research population, and half of it we took to use as our diagnostic group or our training set, and then the other half to validate this model that we built or the testing set. This is a typical spectra that generated from an NMR analysis. So using orthogonal partial least squares, which is an advanced statistical tool to essentially it's a supervised principal component analysis, and it compresses 69 components or dimensions down to a manageable 2 components or 3 components for you to process the data or analyze the data properly. It's based on variance of the different concentrations of the metabolites. And using 69 metabolites, the black dots represent normal and the red dots represent cancer. We're able to see that they lie on different sides of the plot, although there's some degree of overlapping, and you're going to see that with any diagnostic test that it wouldn't be 100% separated. Now, when we took our validation sample and plotted on our model, I'm not sure how well you can see this, but the blue dots represent the validation group, and they lie mostly on the cancer side. This, our statistical software is also able to generate what's called a variable importance plot, or basically summarizing the important metabolites that drive the separation between the normal and the cancer. So using 69 metabolites, ColoDx, the test, is able to detect colonic cancer with a sensitivity of 93% and specificity of 72%. And that's using 69 metabolites and using NMR. Now, choosing the 5 best metabolites, or the most important metabolites that drive the separation, we're still able to get a sensitivity of 90% and a specificity of 50%. Now, for polyps, where the money is in the polyps to detect them before cancer actually develops, using only 5 metabolites, we're still able to get a very, very good sensitivity and specificity. Now, compare that to the current fecal cord blood test, which only has a sensitivity for polyp detection of 15%. Our test diagnostic accuracies are much more superior. Now, we don't see this test as a replacement to colonoscopy. We see it as a precursor or a pre-test prior to colonoscopy, and it allows us to better screen out those that need colonoscopy and basically avoid colonoscopy for those that don't necessarily need it. In Canada, our resources are limited, and as I'm sure all of you know, it would be impossible for every single patient over the age of 50 to get a colonoscopy. Our resources just don't allow that. So our hope is that this test will select out those that need it most and hopefully avoid it for those that don't. The opportunities, so in Alberta, there are 3.5 million people with 100% healthcare access. There is a current single centralized screening program, and so this provides ColoDx or MTI with an opportunity to add ColoDx to an existing screening program in the pre-market test. Now, bringing that to worldwide, in North America and Europe alone, there are 200 million people over the age of 50. And even with an uptake of 40%, that's 80 million people that need colorectal cancer screening per year. Now, we don't know exactly how much this test is going to cost yet, but roughly at $100 per test, that's $8 billion per year. So that's all I have, and thank you.